old, now 46. So yeah, I'm just here just to learn more. It's to see the progress, obviously, from that back in the day, you know, compared to what is now. So yeah.
Hi, my name is Paul. I'm father of a 3-year-old child who was diagnosed with 4 months. She's now 3. She just turned 3 last month, and very happy to be part of this workshop and also in this event. Very important. Thank you.
Great, thank you. Is there anyone else in Vancouver? I'm not quite sure.
I think we're good here.
You're good. Okay, great. And then maybe online, I'll start with Jamie.
Hi everyone, my name's Jamie. I'm a genetic counselor. I've worked in clinical genetics in a hospital for about 10 years, and now I've been with Impact Genetics for 10 years, working closely with Dr. Rasher and this group. It's wonderful to be here again.
Thanks, Jamie. Next, Mary.
Hi, I'm Mary Connolly Wilson. I also come at it from both sides. So I come from a large family with retinoblastoma, and I was also a genetic counselor for 34 years and now retired.
Great. Linda. Linda N. We can skip you. Oh, there you are. Hi. I don't think we can hear you. I can read lips. Your name is Linda. I'm guessing you're the last name you're Mitch's parents?
I can.
Yes. Hi, Lisa. Linda, sorry.
And I'm Mitchell's mother.
Great. Thanks for being here.
Okay.
Hi, Mom.
Robin is next.
Yeah, I'm Robin. I am 45 and a bilateral retinoblastoma survivor. I was 11 months when I was diagnosed. And yeah, this is my first experience in something like this. It's pretty cool so far.
Being here. I'll pass around the mic.
Can everyone? Is the mic working?
Maybe try this one.
Hi everyone, Hillary Racher here in the on-site in Toronto. I provide the genetic testing for many retinoblastoma patients through Impact Genetics. I'm really happy to be here. I've been to a few of these symposiums.
Hi, I'm Tim Cotton. I'm a retinoblastoma researcher here at the University of Toronto. It's my first time at one of these.
Hi, I'm Kyle Perugia. I currently have a son that is going through treatment at Sick Kids. He's 2 years old now. He has bilateral retinoblastoma. This is my first time being at something like this.
Welcome.
My name is Jillian, and I am married to a retinoblastoma survivor.
Thank you. My name is Mark Scanlon. I'm a retinoblastoma survivor. I was diagnosed on my first birthday, and I inoculated when I was 5 when I came back. This is my first being at one of these events. I'm very happy to be here. I'm currently 45.
My name's Jordan, and I'm the mother of a retinoblastoma survivor, Logan. He was diagnosed at 6 months old at the hospital for sick kids and treated by Dr. Galley. He has his left eye enucleated, and he's currently 7 years old and doing well. And he's the inspiration behind the storybook that I wrote, The Extraordinary Eye. And I had the pleasure of meeting Mitch a few years ago at Matthew's office, and yeah, meeting him really brought me to tears because like his mother, I'm an educator, and I remember you, Linda, and your husband very well. And it was around it was in the first year that he was diagnosed, so it was really nice meeting you. And sorry, I don't know why, but seeing Mitch, such a handsome boy, and just a very successful young man, and now hearing that he's going to be a genetic counselor in 5 days, it really makes me feel hopeful for my son too. So I don't know why I'm crying.
You're making me cry now.
But it's just inspirational, and I'll never forget you. And I remember you gave me your contact, and I'll never forget your story about how you were playing pirate with him, and that's when you discovered it. So I don't know, just wanted you to know that you've left a mark with me, and I'll never forget you guys.
Thanks, Celine. Thank you. Yeah, next time we have to have a big budget for tissues. Yeah. That's a good segue though into Mitch and his story. So Mitch and I are co-hosting this Coast to Coast genetics workshop. I'm a scientist here at Sick Kids. I studied molecular genetics. Mitch is supposed to be a genetic counselor and has lived experience of retinoblastoma. We thought we would start with Mitch, actually. So Mitch, before I hand it over to you, we're meant to get through a lot today, but we may end up pushing some of this to tomorrow. We want to go over the lived experience of retinoblastoma and how that relates to genetics. We want to review genetic testing reports and how that information is conveyed to families. And then we want to look at some examples, both online, I'll show them online or virtually, as well as some printouts in the physical locations. You can take a look, scribble on them. And basically today we're just thinking about ways we could communicate that information better. And maybe if there's time, a brainstorming session, or we'll do that tomorrow. So Mitch, I'm going to hand it over to you. And I see there's pirate bones there, so.
Pirate bones are very important. I just wanted to say it's great to see so many young families here today, and Talene, great to see you again as well. Just thinking about the way that Jay was framing the conversation, the importance of community, of seeing someone who's done it before and kind of made it out on the other side can be really powerful, as we're seeing already. So I will try to go reasonably quickly here. The way I prefer to do this is to have the images come up one by one, but with the difference in who's communicating versus who's controlling, I thought it'd be easier just to do it all at once. So starting at the top left, this is kind of my RB timeline. That's a schematic of my family. That's mom and dad. My big brother is the one who's standing up in that picture, and I'm the baby. And it was a very normal family, I would say, for the first 3 years of my life. They had my brother, who's 2 years older. They had me, and for the first 3 years, I was a very good sleeper. I was a very good eater. Everything was good at my well baby checks. Life was great. And they were like, oh my goodness, we have these 2 beautiful babies. Isn't this fantastic? Until one day I was about 3.5 years old, and I was a bit of a dramatic kid, and I liked to play dress up. It was one of my favorite activities. And the outfit of the day was a pirate costume. And so what do you need when you're a pirate? You need a hook hand, you need a bandana or a hat. And maybe the most important part is you need the eye patch. And so mom was helping me put on the eye patch, and she put it on the left side. And the moment she did that, I said the words that no parents would want to hear: "Mommy, I can't see you." And she said, being a bit of a dramatic kid, she didn't believe me. She said, "Yeah, right. Here's my hand, give me high five. You can do it." And I was like, "No, Mommy, I can't see you." And she was like, okay. And so we were very fortunate that my aunt was working with an ophthalmologist at the time. She could take me right into his office that afternoon. It was Dr. Hiscox in Owen Sound, Ontario. And we were very lucky that he'd seen retinoblastoma before, because I had a very advanced tumor in my right eye to the point that I had no vision on that side. And within a matter of weeks, I was taken to sick kids, enucleated on the right side, and kind of treatment underwent very rapidly. And kind of, it was a real thunderstorm of events on that in terms of how quickly that all happened for my family. Interestingly, I know the slogan is "Know the Glow." We don't have any pictures of me with the glow. I don't think that, whether we just didn't take enough pictures or whatever it was, that was, we didn't have that warning sign. And again, we were very fortunate to have Dr. Hiscox, who had seen retinoblastoma before. So made it out on the other side of that. I'm sure my parents were very afraid, very like, oh my God, what is going on? How do we do this? But I would say that I had a very normal childhood. I was not an athletic kid. So, mom didn't want me to play hockey or tackle football. She was worried about, contact sports like that, but I didn't have too much of an interest in playing those sports anyways. I was okay at basketball, but I didn't end up being very tall, so that wasn't very good. Volleyball is kind of out. I find that things coming from above is not a good situation. I did get into music. My family's very musical. Drums were my instrument of choice, again, being a dramatic and difficult child. The Atlanta Falcons logo, that was my high school's logo. I was involved in the student council, things like this. I have the masks on there because I got into musical theater in high school, continuing my dress up theme. And, I'd say my childhood and kind of into adolescence and young adulthood were really very normal. And retinoblastoma kind of took a back seat at that time and came back when I was making decisions about what would I do after high school. I didn't have the level of entrepreneurial acumen that Jay seems to have, and I wasn't going to Broadway, we'll put it that way, in terms of my musical theater skill. So I thought, what can I do? What am I good at? What am I interested in? And I have always been interested in genetics, knowing that this was a genetic disease. From the time I was very young, my parents were saying, there's a genetic thing, but you don't have the gene. They kind of didn't fully understand it, which is a big part of why I want to do this workshop today. But I ended up studying genetics at Western in my undergrad, and then came out of that undergrad with this degree in genetics. And I was like, okay, degree in biology, genetics, this is great. Where are the jobs? And the jobs were nowhere to be found. So I realized I was going to have to kind of specialize more than that. And I didn't love bench work. Anybody who has, experience in their undergrad maybe or something like that, doing bench work in biology, wasn't for me. I wanted to be talking to people. And I, was kind of toying with the idea of genetic counseling, knowing that my parents saw a genetic counselor, knowing that was part of the RB journey, was this genetics thing. And I took the plunge. I, got the prerequisites. I applied to the programs in Canada, ended up interviewing and getting in here at UBC. And that was, as kind of the Cole's notes of my RB journey, I meant to add something that was really meaningful to me is during my training here at UBC, we do an offsite summer rotation for 6 weeks. And during that, I was actually able to go back to sick kids. And I was able to work with the genetic counselors there for the summer. And in hindsight, my skills were not refined at that time. I was relatively early in my training and I still had a lot to learn and a lot of growth to do. But I think that training there was really important for me, just personally as well as professionally. I think it's a fantastic learning environment. The team there is incredible. And now I'm back here again 5 days away. I'm going to have my degree and be on the other side of that. I do have one more slide that I added last night. I don't know if you noticed. So I want to talk about why this workshop. Why am I here for this one and not one of the other ones? It's because of this family, which, spoiler alert, that's my family. And after they, I'd had my care, I'd been enucleated, I had the genetic testing, I had the genetic test report. But mom had 3 questions, and she felt that none of them were really adequately answered by what she was given. The first, and she still asks me these to this day, by the way, why did this happen? The second, will your children, her grandchildren, be at risk? And the third, what on earth does this genetic test report say? A couple of weeks ago, they actually showed me my genetic test report and said, can you tell us, after all these years, what this actually says? And I did. And I don't say that to, belittle. I think they got great genetic counseling at Sick Kids. I'm sure they did. The genetic counselors there are fantastic and have been for a very long time. I think that it really is a product of the reports and, not having something in hand that you can say and look back at and review that after 23 years, you're going to lose that information. So having accessible reports is really important. And that's my intro. So sorry if that went over time. And I'll give it to Helen to talk a little more about the nitty gritty of the genetics.
Yeah. Thanks, Mitch. That's really helpful. Genetics is complex, and I think it was you who told me how to try to simplify my talk. Let's see what you think of it now. Because early genetics, it actually is pretty complex. It's really hard, and when you come from the science side of the world, we tend to get so hung up on this precision, we can't get rid of the jargon. We feel that when we give too little information or we simplify it, we're missing some of the details. So it's kind of it's a struggle, it's a challenge to get the information just right. But before we get into those reports, let's just do a primer. I'm going to start very basic. I know a lot of you already know this, but if you don't, that's okay too. Stop me, ask any questions. But essentially, we're going to start at the beginning. I don't know if that's me or background. Okay. Things freeze. No. Okay. So here showing on the screen is a human being, and we're all made up of tissues that at its smallest level are cells. And within these cells, you find our DNA. And our DNA is packaged up into these things called chromosomes. They're really tightly wound DNA in this sort of X cartoon here. And if you unravel this chromosome into kind of like unraveling yarn, you'll find this double-stranded helical structure, and that's what DNA looks like. And within that DNA are different combinations of bases, which is like the alphabet of DNA. And everybody has a different sequence, and that's what makes us unique. And each of our chromosomes has a complement of genes that do a variety of things. In one of the earlier talks, we saw a slide of Brenda, Lynn Murphy, Ted Dryja, and Webb Kavanaugh, who discovered the genetics behind retinoblastoma. And essentially, this is what they discovered. They discovered the precise location on one of these chromosomes where the gene that causes retinoblastoma lies. So we all have this gene called RB1. And when RB1 is not in its correct sequence, it causes disease, it causes retinoblastoma. So the precise location, that 13q14.2, that's sort of like an address for where a gene lives on a chromosome. And we've, through lots of research, been able to understand that this particular gene controls how cells grow, so how they divide to form that human being. So you can imagine it's sort of like brakes on a car. You need it sometimes to be like push the brakes, and sometimes you need it to go. Sort of the same thing with cells. You don't want them constantly growing, but you don't want them stopping too early either. And we've since found out that the RB1 gene also keeps DNA healthy. And what I mean by this is that sequence of bases, every time a cell sort of replicates, we need to make copies of that DNA. And so RB1 is sort of important in keeping that DNA in its correct sequence and what we say is genome integrity, or keeping it in the right amounts and structure and sequence. And so in this cartoon, we're kind of showing what happens when retinoblastoma initiates. So if we're just looking at this chromosome 13, where the RB1 gene is, it's shown in this is its location, we get an error, or what we used to call mutations. Now you might hear the term pathogenic variant. Basically, in retinoblastoma, we lose both good copies.
keep hearing feedback. I don't know if that's me or, can you guys hear well in Vancouver?
We can, yeah.
Okay, all right. So essentially, when you see on the first chromosome a little red cartoon, that's representing the first error or the first mutation. And the purple one on the second copy of the chromosome is the second one. And so you've lost that gene that controls cell growth. You've lost that ability to keep the DNA healthy. And it's the first sort of step to starting that tumor forming, because you can't control how the cells grow. You're not keeping the DNA in its healthy form. It can acquire more mutations or variants. And so the timing of when this happened is also interesting, because if it happens early on in development, or let's say you inherit it from a parent, then it's going to affect this error is going to be in all the cells of your body. If it happens later on, like let's say the baby's conceived, the cells are growing, and the error in RB1 happens later, like the eye is developing, it might only affect the eye cells, the retinal cells that become the tumor. And now you can start to see when some things might be heritable or non-heritable. Any questions so far? So many genetic counselors that could do a better job than me. So here's another cartoon here. On the left side, we have heritable retinoblastoma. So when somebody has retinoblastoma in both eyes, that's the first signal that this must be heritable, that this RB1 mutation is likely in all the cells of the body. They maybe got it from a parent, or maybe acquired the mutation early on when they were a developing baby. But sometimes you can still have heritable retinoblastoma, but not present with bilateral. We don't know why. Sometimes it's the kind of the way in which the RB1 gene is damaged. Maybe it's not fully damaged, so you only get partial, what's known as penetrance. I like to think of these people as lucky bilaterals. Sometimes you might, oh, Mary's on the call, but she knows, sometimes you might get retinoma. So the tumor doesn't fully form into a malignant retinoblastoma, it stays benign. But what we do know is when the RB1 mutation affects all the cells of the body, we know that these individuals might have a second cancer risk. These individuals may have family members at risk, for example, the next generation. Whereas when that RB1 mutation happens later, it's acquired maybe only in the retinal cells that form the tumor, then that individual has non-heritable retinoblastoma. And this is the majority of people with unilateral, not all, because as we mentioned, some people with unilateral disease might also have heritable retinoblastoma. But most unilateral would fall into this non-heritable. And genetic testing is the way we can distinguish, because you can already see from this cartoon, simply knowing if someone has both eyes or one eye affected, you can't tell the difference in just by how many eyes are affected when you only have one eye affected. So genetic testing is important, and therefore the report is important. Just to make things more complicated, in the last 15 years or so, we discovered that there's yet another way that retinoblastoma might form genetically. And this is through a different gene called MYC-N. This gene is on chromosome 2. It's what's known as an oncogene. So when there's too much of it, it promotes retinoblastoma, it promotes cancer. RB1, when there's too little of it, it promotes cancer. So this is a gene that also regulates cell growth and division of cells. We know that a lot of cancer-related genes are involved in cell growth. I see that Jamie has a chat thing there. Jamie, did you want to say it?
Sorry, Helen, I just, I've written it down. I love the way that you're describing those unilateral patients that have a hidden or maybe a germline mutation as lucky bilaterals. I just think that's a wonderful way to say it. And we're doing a white paper right now, and I'm thinking that might be my title. So thank you.
Fantastic. Thanks, Jamie. So for these individuals, what's interesting, when the retinoblastoma is caused by MYC-N, it's very rare. And in fact, it's cases where we've tried to do genetic testing and we couldn't find the way in which the RB1 gene was broken or there was an error in there, because there is no error in RB1, but there's too much of McN. And so when there's too much of McN, shown here, if you can look closely, one of these chromosomes has like, it's lengthened because there's too many copies of this gene, then this type of retinoblastoma initiates. And so I've just modified my cartoon here to show you that sometimes unilateral, that's non-heritable, would have acquired too much McN. So these are also non-heritable. There's no risk to the next generation. There's no second cancerous that we know of, because it's only these cells in the eye that are affected there with the mutations or the variants. I'll skip over that to just go straight to what does this mean for the patient. So in the absence of genetic testing, we know that bilateral patient has heritable retinoblastoma. We don't know the precise error, and therefore we can't use that knowledge to screen the next generation, siblings, parents. So genetic testing is still important to know exactly the sequence, that alphabet, that's going to tell us what's that family's mutation that we could use to screen. And for unilateral, without genetic testing, we don't know if they have heritable retinoblastoma. We don't know if the other eye might get retinoblastoma. We don't know if there's a second cancer risk. We don't know if parents, siblings, or future offspring are at risk. And we don't know if it's caused by McN amplification or RB1. And so again, essentially this limits our ability to screen that individual and future or current relatives. And so we do genetic testing, and in Canada we're quite well versed in that, thanks to Impact Genetics that Jamie and Hillary represent and are here today. And so most people in Canada, if not all, will get their with retinoblastoma, will have their blood screen and available tumor screen to identify precisely how they're affected by retinoblastoma, by which gene, and what their family's mutation is. And the importance of that is for the life course of the child. So this shows from baby all the way to growing up into adulthood. You can see that genetic testing and counseling can help with diagnosis. That's what DX means. Of the baby, the parents also have that family mutation, or any other children. We can also use that knowledge to screen future pregnancies. And so that's available now, and anything in blue is available now. So what we'd like to do is create more tailored counseling protocols and tools to help that information stay with the family into adulthood. So we're really understanding how families are affected individually and precisely. We can also use the knowledge of genetics to, and what we're working on is how to screen for tumor that might, if someone's under treatment, there's new techniques now where we could maybe monitor or survey if there's minimal residual disease, is what oncologists call it. There's also really new techniques where they're taking fluid from the front of the eye that will tell us if tumor is active or not. So again, we don't know precisely how to do that right now, but there's research happening. And so again, you can see the importance of understanding your genetics to know how this might help current or future treatment down the road. And of course, precision care in future for the next generation. Right now, we're using the family's genetic testing report to screen future pregnancies, or so you can do prenatal diagnosis, whether that's through amnio, or there's new techniques that are non-invasive, where they're finding little bits of the fetal DNA in the blood of the mom and testing that. So that's called non-invasive prenatal diagnosis, NIPD. And then there's also pre-implantation genetics. So some families may want to go through in vitro fertilization and screen out the family mutation from the embryos that are implanted and brought to term. So there's a number of ways that genetics can help care, both now and in future. And so that's why it's important that we're conveying that information completely and in an understandable way. So maybe we can have a little bit of a discussion now on how do patients receive information about genetics? How well is it working? Can we do better? I know Hillary had some examples. I put the screenshots here if you want to come up and walk us through them.
Yeah, I could.
Is that okay?
Yeah.
And maybe we can talk about how we do better. So
Yeah.
there you go. So we just click through, or I can click through for you.
Yeah.
Yeah.
Yeah, great. So, something that Jamie and I do on a regular basis is provide genetic testing for both the patient with retinoblastoma, but also their family members. And on occasion, we do get prenatal samples to support different stages. Now, there's a lot of information here. We were talking earlier about conveying things in a tangible manner.
And some copies too.
Okay, yeah. So there is a lot here, and certainly these reports are typically directed to the physician caring for the patient. So sometimes not the simplest language used, and this will be a great opportunity to take some of these more challenging to digest and convert it into what Helen wants to accomplish in this session, which is more tangible materials. So you're not gonna be able to read this, but this, I'll take you on a journey through how I construct and what goes into a genetic test result for a retinoblastoma patient. The one that Helen has up right now is an instance where a patient did undergo a nucleation, so we actually have access to the tumor. So there are two results here. There's a genetic result from the tumor itself, and we also received, for this patient, we received a blood sample. So earlier today, we learned about the term, I think it was Bruce, described that the rest of your body, you usually refer to that as the germline. It's also known as constitutional cells. The blood is representing the rest of your body. The tumor results are specifically DNA extracted from the eye tumor, the retinoblastoma. So you can see, I guess just the general layout for a genetic test report, like any other report, starts off with some information about the patient themselves, their age, their diagnosis. The lab gives some identifying information. We need some identifying information to take the samples through the lab and to keep track. There's also a location for physician information and all the contact information. And the more interesting section is the middle here, where we describe the test results. So we always begin our reports with an overall conclusion for the test results. So in this case, the patient, through our testing, we identified that this patient has heritable retinoblastoma. And as a result of that, we've been talking about the risks for potential increased risk for second cancers or second tumor in the eye. And then in the body of the report, we list exactly. We had the tumor specimen in this case. This patient's tumor showed a very specific, there's a lot of, for the genetic counseling and anyone who studied in molecular genetics, you learn about how to describe a variant, a mutation, a pathogenic variant. It's complicated when you look at it for the first time, but it's zeroing in. Helen described, the shape of a chromosome down to the nucleotide level. So our report describes precisely that precise location inside of the RB1 gene and how it was altered. So that's what's been described as allele 1. We have 2 copies. Helen instructed us at the beginning, got 2 copies of the RB1 gene. So allele 1 is referring to the first copy, and it has that alteration. The other copy in the tumor has something called promoter methylation. RB1 can be damaged in a lot of different ways. And one way is a specific region that regulates the beginning of the gene. You can have abnormalities in the methylation profile. So we've covered the 2 hits in the RB1 gene. As Helen described, if you damage both copies of your RB1 gene, as the beginning stage of the retinoblastoma cancer. We've triggered the cancer form. These are the two signatures. What we also found when looking at the DNA in the blood, which represents the rest of the body, extracted DNA, you'll see if you could, probably in the printout copy, the same variant was found that you identified in the tumor. In this instance, the patient was born with this alteration, and all of the cells of their body represented in the blood. And so that's why we refer to this case as a heritable retinoblastoma. Describing in the body of the report is the interpretation section. Goes into looking in the literature. You know, a lot of my job as a molecular geneticist involves digging through what we know about a particular variant, so we can decide whether it's the cause. Is there enough evidence to suggest that it is damaging the RB1 or not? In this case, there was enough evidence. The evidence is listed in the interpretation section for both of the variants that were found in the tumor and the consistent variant that was found in the rest of the patient's body. In the section below the interpretation is the guidance and recommendation. Kind of goes back to what Mitch was saying earlier, his mom's questions about what does this all mean. In this location, we talk about, you know, what does this mean? For this patient, you know, this means that genetic counseling is really important to discuss the implications of this finding. This is heritable retinoblastoma, so there is risks for potential second cancers, and there's risk for the family, and that's what's listed in the table below. So parents, in this circumstance, the parents could, as Helen described earlier, parents could be carriers. And in this example, I don't know if I included that, but it was considered an isolated case. Parents weren't presenting with retinoblastoma. It doesn't mean that the parents don't carry. There's something called I'm getting into jargon probably, but inside of sperm and oocyte of the parent could carry a population that carried the RB1 variant. So there, even if the parent doesn't present in this circumstance, there is a 5% risk. Could be in their sperm or oocyte. So they could potentially have other children. Siblings in this case too, there, of course, if the parent does carry in sperm or oocyte, there is a risk that another future sibling could inherit. And the largest risk, of course, is for this patient's offspring. This is heritable retinoblastoma. That means all the cells of this patient's body carry the variant, and therefore their offspring have a 50% chance. The second page is for those that want to learn more about the methods.
Yeah.
And I won't go into that right now.
Thanks so much.
Yeah.
Any questions from the group on the report or anything? I guess one thing we should say is the reports are generally made for the scientific community. So this is the report your doctor would get from the genetic testing lab. It's the report that the genetic counselor would interpret. And sometimes genetic counselors also provide supplemental information, which I'll show you in a moment, to help decipher this. But just to pause here, what are your first thoughts on the genetic test report? What do you like or not understand? Or love? Well, first impressions. This is just. Anybody from Vancouver want
to? I think we're kind of looking around, kind of confused glances. Kind of like, what's going on?
It's a lot of information. it works well on the medical and clinical side. All this information has to be there according to the genetic standards of reporting, right? The question is, how do we take this information and distill it down to what the patient family want? Mary, you have a comment?
Yeah, I just wanted to say, I mean, you know, in reality, and being from both sides, you know, um, what you need and as a genetic counselor, what you can give are two different things. So, I mean, when we, you know, when you see a patient initially, that patient's first diagnosed. So, you know, the parents are very upset. They're not taking a whole lot in, simply because the emotional impact of that does not allow you to really take in the knowledge that you need. And then the second time you might see a genetic counselor is when your test results are back and you're still being, you know, it's like a great big bat to the head. You know, all of a sudden you realize, oh my God, yes, this is genetic now. You know, I may be at risk, others may be at risk. What does this mean down the road? There's 1,000 questions going through your head while a genetic counselor is still rattling on at you about what this report means. And it's not that they're not listening to you. I mean, we know as genetic counselors that they're probably not hearing a word you say because there's all this other stuff happening to you in your mind. So ideally, what would be great is if you had, if everybody, you know, who had a genetic disorder had 4 or 5 sessions. You know, you get your one at the time of diagnosis, you get your next one after the testing, and then 1 or 2 years after that, maybe you get another session to review what you didn't hear in the beginning. And then, you know, there's also, you know, maybe when the child comes into their teen years, what do I say to my teenager now? And so maybe you come in for a counseling session, then the teenager comes in for a counseling session. That person then comes back for prenatal counseling. All those things should be done. But in reality, in Canada, we don't have that ability to do that because, I mean, most genetic centers across the country have wait lists for counseling sessions. And so in reality, you might get it, you're lucky if you get 2 sessions, the one at diagnosis and the one after, you know, you get your test results back. So they're not the ideal times. And like Mitch said, his parents, you know, never truly understood. And I can see why, because you weren't at your optimum when you got those. None of the patients that genetic counselors see are really at their optimum time to be hearing all this information. So can we find a better way to do that, or can we give other things to help that, you know, to help parents and individuals themselves absorb it? That's what we need to look for, because I don't think we can change our health system to create what we need. That's just my take on it.
So I just wanted to share my experience. I was actually just going through some old emails. Sorry. I just want to share my experience. I was going through some old emails just to kind of have like to remember what happened back then. And I remember 6 weeks after is when our results were shared with us. And the day that my son had his surgery was the day that the genetic counselor came and wanted to do like, kind of idea, like a family tree, like what cancers or other ailments our families have experienced. And it was during surgery. So I'm like, this is not a good time. Like, I'm sorry. Like, I was just frazzled, but that was the only time she had to do it. So I did it with tears and like, it was just not a good time. So I think really like taking into account, like that we're human beings and that, yeah, okay, I understand science and medicine and everything, but let's just kind of get over the hump first and then we can discuss all the specifics. But I think that piece was missing, the empathy piece. And also the lack of like having, like this is a workshop about how we can like create educational tools. So being an educator, I think having something tangible, like, you know, giving out a handout, like a little pamphlet, like here's a glossary of terms. This is what your child is most likely to have, but once the genetic testing comes through, I think that's when I would give out that pamphlet of terms. Like I wouldn't give anything out before that. But to this day, I still have questions about what is mosaicism? Like my son is 99.99999% non-heritable RB, but there's a very slight small percentage chance of mosaicism. And we're going to talk to your son when he's ready to have children and get them tested because there's that small risk. But what does that even mean? Like, what is mosaicism? Where did it come from? Like, what does this mean? So I think even having like just basic stuff, like phonetically even having it spelled out in brackets and like a very brief, like layman terms type of definition, like kind of like our cup of tea, but in the way that we would approach it, like, you know, in this format. So I think that's important to have, just like as to use as a tool, as a guide, because like I said, to this day, I'm still confused. Like I emailed Dr. Gelly so many times. I felt so bad. I'm going through all of that, like maybe about like 5 to 10 emails just to her, Dr. Sammy, and to our genetic counselor at the time, and like not really having an idea of what's going on. And even like I looked at the date he was diagnosed in 2017, up until 2019, I still had questions, just did not know what was going on. So, and now I'm thinking, I'm looking back, and I'm trying to remember, like I just wrote down all the stuff, like I want to go home now and see, did he have RB1 version or the other? Like, and then I always like, and then the anxiety kicks in too, like, did he have this, like, cause they did take his eye out, so they had the tumor, did they test it against his blood? And I'm constantly going back at that and asking my husband these questions. So I really think anxiety is really high for these patients' parents, especially, and I think that finding the right time to do this, to have that conversation, the right time to give that pamphlet, I think all of that needs to be taken into account. Yeah. Sorry.
No, it's important. It's timing is a big thing. I don't think we're gonna solve today here, but it's noted. We can solve the educational tool piece maybe partially with the work we want to do here. Jamie, do you have a comment or?
Yeah, I have mostly a question, and I think thank you for sharing that experience. And I think I'd like to let you know that you are not alone. And I'd say the majority of even genetic counselors that don't specifically work in retinoblastoma, it is much more complex than a lot of other conditions. So I just want to let you know that you're certainly not alone. Ocular oncologists, even the best of them, still struggle with mosaicism. So my question for you is, and we're actually, Dr. Racher and I are in the process of doing a white paper on unilateral retinoblastoma. In your opinion, if you were to think about an ideal way to get the information like reviewed with you, would it be a newsletter? Would it be a video? Would it be a one-to-one? Like what would resonate the most with you?
I think a one-on-one would be really good because then I can ask questions right away in real time and get back. We had this form, like literally the same thing mailed to us, and then like the same thing emailed to us like later. Yeah. And I didn't know what I was reading. I didn't understand it. And you know, not for nothing, I have post-secondary education. Like I'm a teacher, and I didn't understand it. Like I would, now that we have ChatGPT, I would probably take it, upload it, and have AI tool read it back to me and say, read this to me in layman terms. And even some people don't even know that they have those tools available to them. But to have something like, you know, ready, I think that's really good. Yeah, and I think in real time, yeah, just to say, like I said, like to have questions answered right on the spot, I think, so that there is more anxiety being built up.
Thank you. Yeah, it's good. I think it's good to keep in mind, Helen. I think just through, you know, years and years of trying to create written documents to explain like the results of a negative genetic test with residual risk has been so difficult. And so I'm wondering if literature all we want, but perhaps dedication to time with families or even videos as a second best might be a better way to go.
I think you're right. I'm going to move to some of the options other people are experimenting with to help convey this information. So Hillary nicely sent me some nice, I'll share these after if that's okay with the group. The reports, there's no personal information on those. But some places use summary letters. So these are examples of summary letters provided to me from one of the sick kids genetic counselors. So sometimes they send these letters along with the report to try to break down some of the information. Again, it's a written thing. It's not, people with different familiarity with this kind of information may benefit from it. Others may still find the diagrams a bit too difficult to understand. I'll pass these around to, so you can take a look on your own. And I think Vancouver has some of these as well. This is like one for each, yeah. And some other evidence-based approaches, and I think the impact report does this. So essentially, other places have altered their genetic testing report to include an interpretive summary section. And I think that's what we have. There's a way to make it maybe more plain language. But essentially, if we look at the impact report, this interpretation section that's written in a little bit more medical terms, it might be simplified. So the regular genetic testing report would just have a simple section. So we can discuss the pros and cons of that. Others have used a user guide. So if we're going to keep the report the way it is, and we want to teach people how to find the information, it might look something like this. So that alongside the report that Hillary showed us, have a user guide that tells us here's a glossary of what things mean, here's how to find the information you want, and anything else. Then there's a whole other approach of the comic books, and these are sort of supplemental, sort of looking at educating groups into sort of like the information I gave you earlier. This example, which I emailed to everybody, is a draft booklet created in Kenya by retinoblastoma parents and survivors. And in that setting, there's no genetic testing and there's no genetic counseling. So essentially, people only understand heritability in terms of how many eyes are affected. So in that case, people with bilateral retinoblastoma might be told by their doctor that they have heritable retinoblastoma, but we don't have a way to screen their family members. And those with unilateral, because of the absence of genetic testing, really they're all told, you know, bring the next baby for a checkup and we'll monitor the eyes because again, there's no genetic testing. That's changing. But in order to simplify some of that uncertainty, the group made these cartoon booklets to try to explain everything from what are cells and genes and DNA to more complex family trees. And we have it in English and Swahili. I sent you guys the English version. And then also from the African setting, here's a more complex comic book that's geared to children. And this actually goes into really complex genetic technology, new sequencing methodologies, and it makes it sort of an action hero booklet. So kids can read that. And I emailed you all that as well. That's the Botswana Ministry of Health. So that's not quite individualized explanation of the report, but a supplemental educational tool that might make the reports a little bit easier to understand, or at least the concept of genetic testing. And then there's the idea of having a whole new plain language report. So this one would be a supplement also to the report, like the one from Impact, but it would be redesigned. And here's an example of one from Australia. And they work with patients to make a simplified report. I'll also hand those out. There's a bunch of different templates, but just pick one and take a look. Essentially, it's all the same information that was in the impact report, but formatted a little bit differently so that it's only the essential information. You know, like the impact report, we have the patient's names at the top, the reason for the test, a little bit about the test, the patient's result, simplified, and then the idea of the interpretation of what that means for inheritance, the recurrence, the recommendations, but really simple language. What happens next? Your genetic information that you go back to. And then this interesting thing of the QR code where you can scan that and get even more information. So we could experiment about with making our own version like that. And I'll also remind you that in one of the past cup of tea discussions about survivorship, a lot of RB survivors mentioned not having a tool or some sort of backup information to show their healthcare providers outside the RB team to justify why they don't want to do that extra CT or the dental x-ray, or perhaps you could incorporate something like that into a plain language report, which serves the purpose of simplifying your genetic results, but also helping you advocate for your health outside of the RB team. But all different ideas. I'd like to hear your thoughts on, based on the summary letters, plain language reports, supplemental comic books. Celine says she likes the summary, this plain language report, or the summary from Sick Kids. Summary from Sick Kids, excellent. So the visual part of the summary is really helpful. Other comments, questions? And I'm gonna ask the volunteers in the back how much time we have. We're cutting in a couple minutes. So we're gonna think about these things. Everybody's sort of trickling back in the room. I'll send out the electronic copies as well, because tomorrow we're gonna brainstorm what our preferred option looks like. Make sense? Can you be online? No. Okay, well, I can remember us. I know where to find you. Any comments from the group online or in Vancouver? Yes, Jean. Hi, Jean. Oh, I skipped you in the discussion. You never introduced yourself. I'm so sorry.
Hi. Yeah, thank you. I'm just listening to some of this. I'm getting quite excited about some of the graphics coming out of Africa. And it also, one of the things that I found working with Impact Genetics was that people who had had unilateral disease, particularly, once they had gotten through all the surgery as the family, it was almost like they put it behind them, and they forgot about that this genetics was going to follow them through their lives. And so I was just thinking that a comic book sitting on a child's bookshelf would be something that would catch their attention, and that they would have that going forward. I know, like, years ago, Joanne Sutherland at Sick Kids had 3 different versions of the report that she sent out, which was very cumbersome. But often those got lost, and it was hard to find them again. So this is a great topic. I just love where this is going.
Thanks, Jean. So hopefully that stimulated a few thoughts, and overnight we can take those. I see the other groups coming back. So we'll to be continued tomorrow, unless there's like last thoughts from Vancouver. No?
I don't think so. Just to say that really great discussion, and I definitely echo everything that's been said by Mary and Talene and everybody else. It's a really heightened time to be getting the news, and I think, you know, if we can't control that, it would be great to see what we can do with the resources.
Thanks, Mitch. Really appreciate that. Okay, to be continued tomorrow.